Thank you. Hello and welcome to this webinar showcase on UC Ventures for COVID mitigation. I'm Victoria Slivkoff, Global Head of Innovation and Entrepreneurship for the University of California System. If you haven't already, please register for part 2 of this showcase, same time on Wednesday. I will post the link very shortly in chat. Today, you will hear from 4 UC Ventures. Each will present for 10 minutes, followed by a 5-minute for Q&A with our expert panelists, Doogie Graham and Christian Bramer. Doogie Graham is the Chief Transformation Officer for UC Health, which works with all 6 UC health systems to improve patient outcomes. Christian Bramer is the founder and CEO of Benefunder, which works with private institutional family offices on their philanthropic and impact investing interests in emerging scientific breakthroughs. If audience members have any questions, please use the Q&A function and we'll try to get to those live, or the presenters will answer them in the Q&A function. Now I will turn it over to our first presenter, Dr. Albert Shao and Dr. John Exterio-Chilias from Arterys, UC San Diego.
Thanks a lot. I think John will present the slides.
Great. Hello, everyone. Can everyone see my slides?
Yes.
Perfect. I want to start off with saying thank you for this opportunity. My name is John Exterio-Chilias, and I'm the CEO and co-founder of Arterys. And I have on the line with me Dr. Albert Shao, who's one of our co-founders. And just to start off with our team, we have a very experienced team that with a lot of diverse backgrounds in healthcare. I won't go through the entire team, but just want to mention that we play in the medical imaging space. So having experience with dealing with medical imaging, both from the hospital side as well as from the vendor side is really important, and especially having we have the team that has both sides as well. So with that, maybe I'll start off and give the ball to Albert and talk about what he's doing at UCSD and how it connects to Arterys.
Yeah. So I have to say I am one of many labs in the entire UC system centered in radiology, developing a lot of AI technologies for imaging, as it's a huge area of opportunity. We started off as a company, Arterys, developing a technology that I initially wrote software for while I was a resident at Stanford, but I'm not unique. There are many of us across the UC systems at each of the different campuses, UCSF, UCLA, and UCSD in particular, but others as well, Irvine and others. I do a lot of training of our future generations of physicians. As you can see, go ahead and click next. But in particular, we've also been focusing on AI for X-ray and COVID-19, even before COVID-19 started, with the idea that AI could improve our ability to diagnose disease and sort of level the playing field in a way that take some of the expertise required for interpreting medical imaging a little bit out of the equation, because it becomes really challenging as the volumes of imaging studies increase rapidly to keep up and do really well for all of our patients. We're reading 400 studies in a weekend, for example, and it's very difficult to maintain quality without some AI assistance. So go ahead and move to the next slide. This is an example of what our AI algorithm looks like currently for COVID-19. And just keep in mind, of course, we have many other algorithms here. Our initial results from our first publication were quite good on public data, trained only on public data. But as we accumulated data from patients with COVID-19 within our hospital at UCSD, we've been able to use that data to train the algorithm to learn even more precisely what images at UCSD look like and what sorts of range of pathologies we have to discriminate between. It's not usually just COVID-19 versus something or versus nothing. It's usually COVID-19 versus something else. And so that's an important facet of how AI development will continue to go as time goes on. Can you go to the next slide, John? It's important not just to think about it as a adjunct to diagnosis, but what else can we derive from it? We're doing both the tasks of quantification and localization and detection of disease, but ultimately we want to be able to predict survival, hospitalization to triage patients to where they need to go, whether they can stay at home or whether they need to be in the hospital because they're at high risk of requiring mechanical ventilation and mortality. All of these algorithms require industry partnership for us to be able to cross that last mile and get it into practice. And that's where Arterys comes in. That's where GE came in for our industry sort of co-development of an AI system for automating cardiac MRI, which has been the last project that we spent most of our effort on. But COVID-19 is now central. So John's going to pick up from here and talk about the last mile problem, essentially.
Great, thanks, Albert. And just to give you a little background on Arterys, we've been around since 2013, and our vision is really a world where clinical care is data-driven, intelligent, and available to everyone. And what we're doing is really disrupting the medical imaging space by bringing AI, clinical applications, and cloud to try to reimagine the way medical images are used to treat patients. We started off in cardiac imaging, specifically cardiac MR, and since then have brought on additional applications in oncology, specifically lung nodules, and really broadened our platform. And I'll talk a lot about our platform and specifically marketplace and what we've done in that space. I want to mention there are a lot of other companies building specific AI solutions and for COVID-19 or even broader than that. And where we come in is to really try to aggregate all these AI models and applications into a single platform and a single experience for the end users, which right now is a challenge. I will mention that we are already deployed and live at three UCs, UCSD, UCSF, and UCLA. And we're looking to kind of connect the dots across all campuses, actually. Just to give a little background in terms of what market we play in. Currently, we're kind of on the right side of these bubbles in terms of marketplace. It's about a $2 billion market segment where that encompasses AI algorithms and conventional clinical applications. And I will say that there's this other side of medical imaging, which is mostly on the technology side in terms of PACS, which is primarily for viewing and for storing medical images. And what we're seeing is these two bubbles are slowly becoming one. And Arterys is really built for this next phase, next generation PACS, to be able to provide not just the viewing and storage, but the additional AI, additional clinical workflow to make the end users more productive and more accurate ultimately. In terms of what's the challenge in medical imaging today, just irrespective of AI or COVID, really we know that the current infrastructure isn't built for the issues that radiologists specifically are facing. So one is burnout. We know that radiologists are seeing many more exams per year, and the number of images per exam is growing as well, and that causes misread rates. And we feel that AI is one way that we can help solve this. It's not the only way, but AI combined with the right infrastructure and the right clinical workflow together can truly drive efficiency, productivity, and accuracy. So one thing that we've seen is a shift of, I would say within the last 5 years of trying to bring AI into the clinical workflow. And it hasn't been easy, and we're still, I would say, at ground level. And part of that is because trying to change that last mile that Albert talked about of integrating AI, it's a challenge because you're dealing with legacy infrastructure, and you're dealing with health systems that aren't haven't been historically open to the cloud and haven't been historically open to sharing of data. But what we're seeing, especially now with COVID, is this acceleration of using digital solutions and data to help drive clinical decision making. And for example, just the fact that we're all on a Zoom call right now, and I'm seeing it across multiple health systems, is like people accepting cloud, accepting this remote world that we're now in. The teleradiology is another area where that will continue to grow. So what we're seeing is AI can help make things more streamlined and help address some of these pain points. Now, how does Arterys fit in? So we've built truly kind of an open ecosystem, and we call ourselves open as opposed to some of the other marketplaces out there, because we are catering to many stakeholders within this ecosystem. So first off is we're developing our own clinical applications, but we're also bringing in content from outside to help bolster our offering. And part of that is through through UCSD and what Albert's lab is doing, but also at other UCs. We're working with all these developers that are kind of creating great content, and we want to be able to integrate that into a single unified platform. And that's kind of unique in the space, because right now there's point solutions, but we know that IT in each one of the UC campuses is strained. They can't manage to integrate all these point solutions across all campuses. So where Arterys can come in is bring this unified platform across all UCs for secure data sharing, but also to share AI across the different campuses. And that's, again, our key value proposition of how we can take this single platform, and through our distributors, GE, Siemens, and others, we can bring all that content to our end customers. One thing that I want to highlight is we decided in March to beta launch our marketplace specifically for COVID-19. So it's actually live right now. You can go to arterys.com/marketplace, and you'll see all the COVID-19 solutions we have specifically for X-ray and CT, where we have algorithms that quantify the extent of COVID-19 and we can measure progress. And we hope to go full the general availability by the end of the year with more broader set of applications. But we wanted to really have this available for free for anyone in the world to actually go and use this to test, but also to actually integrate into the clinical workflow. And we've done this now on a global level, starting in North America.
Asks, because we just want to show that one last slide because we're out of time. If you could.
Sure. And then just lastly on the revenue side, it's built primarily on clinical applications and services. So, and this is our last slide. One thing I want to mention is, in terms of our ask to UC, is one is a broad level support across the UC campuses. We have it kind of in areas, just at 3 campuses today, but I would like it to be broader than that, as well as facilitation of what Albert's doing in his lab and deployment across all UCs. And lastly, we do have $5 million left in our Series C financing. And that is, that's all.
Great, thanks, Albert and John. Now I'm going to turn it over to Doogie and Christian to kick off on the questions.
Yeah, hi, this is Christian. Just a fantastic job, and congratulations on all your progress. I mean, what you're doing is so neat and is clearly where things are going. I was just wondering if you could elaborate a little bit on your capital raise strategy. I see that you guys have raised, it was like 71 million so far, and you have a Series C open right now for 5 million. Can you just talk about the nature of the capital that's come in so far, and then talk a little bit about you know, what type of milestone or evaluation inflection point that this next round will get you?
Sure. So up until now, we just closed our Series C, which was at the $28 million round. And since then, I guess before the Series C arrived, we'd used that capital to really invest in the platform and in the technology, and making sure that we bring in content from our develop our own content, but content also from our partners. So moving forward, how we're going to leverage this additional capital is to kind of focus on the commercialization side. So primarily Europe and US, broaden our kind of integrations with health systems, and work with what we call ecosystem partners. These are large vendors in pharma, medical device, and PACS vendors that need a marketplace, and need solutions like ours. So we'll be allocating that capital to just to connect with those sort of partners. And in terms of additional capital, that extra 5 million would just accelerate these integrations and time to market, because we know that it actually is very costly building these solutions, and even that $5 million will allow us to bring on more developers just to go faster and to get to market, get these AI solutions in the hands of what we call our centers of excellence this year, next year, so we can show evidence. I think evidence is the piece that's lacking in the industry that we want to really focus on.
Great. Do we have another question for Doogie?
Hey, John and Albert, thank you so much. That was excellent. I think if I take it more as my kind of as a system-wide COO type perspective, how are you thinking of the, you're obviously in sort of, San Francisco, San Diego, LA at the moment. What's the kind of expansion? How are you linking to the kind of EMRs in terms of the Because clearly our providers want as frictionless an experience as possible. How are you kind of pushing that? And then, what's the next steps?
I think I can field it a little bit. So I have colleagues across the UCs and amongst the radiology departments. We've initiated a UC Radiology AI initiative that to try to figure out ways in which we can communicate with each other, share data, develop algorithms together. And I do a bi-weekly call with some of my colleagues at Irvine and UCSF. The big issues are really clarity on MOUs, clarity on material transfer agreements, some streamlined process for facilitating that. I see there's two sides of that. There are the legal agreements, and then there's the infrastructure for communicating and sharing that data and algorithms in a way that's secure. And at the end of the day, everyone seems to be most comfortable with protected health information locally stored, while de-identified image data can cross between institutions. And that's sort of the, interestingly, that's the framework that Arterys has developed. So that's what we're using. But some of these processes in the UC system are kind of slow because they, they're governed at the individual campus level.
Great. Thank you, John and Albert. Now we're going to move on to the next presenter, Dr. Dan Kamei and Daniel Bradbury and Yushen Du from the Kamei Laboratory at UCLA.
All right. Good morning. My name is Dan Kamei, and I'm a professor of bioengineering at UCLA. Today, I'm going to be talking about our work in developing a novel point-of-care device for the direct detection of the nucleocapsid protein of SARS-CoV-2. As many of you know, like some other viruses, there is a presentation and incubation period associated with SARS-CoV-2 infection. Moreover, many individuals are asymptomatic, further spreading COVID-19. Therefore, a pandemic strategy has been developed where we test, quarantine, and trace. And although testing is a key component of the strategy, it's currently expensive, slow, and not widely available. So our approach to address this problem is to develop a diagnostic to detect the initial infection and to do so in a way that is cheap, fast, user-friendly, and sensitive. And as mentioned on the previous slide, we want to detect the nucleocapsid protein of SARS-CoV-2 at home, in mobile clinics, or anywhere at the point of care or POC. So our product is a modified lateral flow immunoassay, or LFA, that combines a colorimetric signal enhancement reaction with a novel engineering casing design to achieve sensitive detection. A common application of the LFA is the over-the-counter pregnancy test, where capillary action drives the flow of fluid on paper, and there's also nanoparticles that are bound to antibodies that seek out, bind to the target, and then bind to lines printed on the paper. So LFAs belong to the general class of category of immunoassays, along with the lab-based enzyme-linked immunosorbent assay. Another general class of diagnostic assays involves nucleic acid detection, primarily focusing on the polymerase chain reaction. So now if you focus on LFAs and think about its advantages, it's cheap, it's fast, it requires minimal training and equipment, but it suffers from low sensitivity. When we think about these lab-based tests, they are expensive, they require training, multiple handling steps, and equipment, but has a key advantage of being highly sensitive. So what we wanted to do was to combine all of these advantages together. Basically, take all these green check marks and put them into one. And if we think about the enzyme-linked immunosorbent assay, it achieves its high sensitivity through a signal enhancement reaction. And therefore, we decided to incorporate a signal enhancement reaction into our device, which is schematically shown here, where you take the sample, you add it to the sample pad, it resolubilizes components on the conjugate pad, we get binding to occur at the detection zone, and then there's an absorbent pad that provides the wicking power to drive this fluid flow. The user will then just press a single button, causing another fluid to flow through the strip and enhance the signal. So a line that previously was not visible to the naked eye becomes visible to the naked eye, further enhancing our sensitivity. So I'm not going to get into the details of our product, but we filed the provisional patent on August 19th, and we've begun to do a competitive landscape analysis of other point-of-care detection devices that are commercially available. So if we first focus on the Abbott ID Now, it's quick, it uses a swab sample, but it's expensive, making it not amenable for at-home testing. However, because it detects for the RNA from SARS-CoV-2, it can detect the infection within the first week. Now for the Quidel and BD systems, they're also fast. They use a swab sample. They're cheaper than the Abbott ID Now, but more expensive than the LFA because you have to purchase a reader to read their LFA strips, not making them amenable to at-home testing. However, these two systems detect for the nucleocapsid protein, and therefore they can be used to detect the infection within the first week. This also further justifies our choice of biomarker. Now you might be wondering, well, what can we use to detect for the disease at home? Well, for that, there are serology-based LFAs, which are fast, they use a blood sample, they're cheap, just like a pregnancy test, and so they can be used at home. Also, it may be argued that blood is the preferred bodily fluid for at-home testing, since currently swab samples are performed by a healthcare provider or by the individual as the healthcare provider looks on. However, for the serology-based LFAs, they do not detect within the first week of infection, since they detect the antibodies that the individual mounts against the virus, so some time has to pass. So our product, our engineered device, is going to be cheap, use blood as a sample, it'll be fast, and it can also be used at home and detect within the first week of infection, because we're looking at this nucleocapsid protein. So the team that's been working on this is as follows. There's myself. I've been working in LFA-based devices for over a decade. I co-founded a startup, Phase Scientific, which specializes in point-of-care devices, as well as sample preparation methods. One notable accomplishment was the recent commercial launch of an extraction kit for RNA from SARS-CoV-2. Dan Bradbury, who's with me here today, is a 6-year PhD student and has had multiple successes while he's been in the lab. Ren Sun, a professor of pharmacology and bioengineering, an expert in virology and structure biology, who's developed platforms for vaccine development and engineering proteins. Yushen Du, who's also with me today, is an adjunct assistant professor of pharmacology who's established high affinity monobody binders for SARS-CoV and SARS-CoV-2 and has developed a high throughput system for RNA viruses. And then last but not least, Ben Wu, a professor of multiple departments at UCLA, an expert in materials engineering and medical device development. He has over 30 patents, co-founded several UCLA medtech startups, including Phase Scientific. So where is the team now? Well, before I get into the data, I first want to highlight that for SARS-CoV, the virus responsible for the outbreak in 2003, it shed its nucleocapsid protein into the bloodstream, and the relevant concentrations were between 100 picogram per mil to 3.2 nanogram per mil. And a similar range is expected for SARS-CoV-2 due to similarities in viral pathogenesis. Now, if you look at our data and start with just a conventional LFA, you can see that we detect down to 10 nanogram per mil, which is outside of the relevant concentration rate. Now, with our current technology, which has not yet been optimized, we get down to 1 nanogram per mil. So we're already within the relevant range of concentrations, and we're confident that we can drive this down even further. However, the current setup is not as user-friendly, takes 40 minutes to result, and although this data is in serum, which is nice to see, it's not whole blood. So from now until December, what we like to do is further drive down the detection limit, make the user steps simple, just 2 simple methods of just adding the sample and then pressing a single button, reduce the user time to 20 minutes, handle whole blood samples, and if other bodily fluids like saliva become interesting for nucleocapsid protein detection, start looking at those as well. From December to March, we want to do a small-scale head-to-head study between our device and the gold standard and further optimize our device as necessary. And then after March, do some large-scale manufacturing and clinical trials. So the ask right now is for the first 7 months. We're asking for 150K, 100K for the salaries of the individuals developing the device, as well as supplies for the device. And then $50K for patient samples, the supplies for the expensive lab-based tests, and then the personnel to do the head-to-head study. We're also asking if there's access to other patient samples and personnel to run the head-to-head studies. Down the line, we're thinking about casing and LFA manufacturers, as well as regulatory and distribution expertise. So here's my contact information. I'm Dan Kamei from UCLA, right? And I like to be contacted through email. So it's kamei@scas.ucla.edu. And this concludes the presentation, and I'd be happy to answer any questions. Thank you.
Hey, thanks, Dan. Okay, I'm going to turn over to Judy and Christian now.
Great. Hey, Dan, great presentation, great job. Looking forward to this getting commercialized, and actually relates to my question. Can you expand a little bit on what you see as the path to commercialization and kind of the inflection points along the way that might also tie into capital requirements?
That's a good question. So as far as the business side of things, I don't think we thought too much about it. But I feel like, we're looking at home tests. We're trying to make something that becomes very widespread, where asymptomatic individuals could use it as well. The business angle of this, I have not really thought through.
Thank you.
I guess that's part of the ask too.
Yeah.
And that's fair, Dan. And again, I think where you're at, definitely, if I think of all the work we've been doing with Oh My Garner and others, your UCLA and all the other sort of lab directors, this fits firmly in the sights of where we want to go in terms of both the turnaround time, the ease, the scale. So I don't have any specific questions, but this is exactly in line with where we know that we need to get as the campuses look to open up. Our current 7,000-8,000 tests we can do a day across the whole UC system just won't cut it, and particularly not at a couple of $100 a test, and sometimes many hours, if not a day, for turnaround. So this really is where we want to go. So thank you in advance for this. This is excellent.
No, thank you. Yeah, we did just a quick cost analysis, and this is based on purchasing for a lab. So definitely the cost would go down if you scale it up. But it's like $1.50 roughly per device. So definitely could get widespread. Yeah.
Definitely, Dan, and that's got to where the numbers have got to pan out to. As we look at continuous testing, and particularly the asymptomatic issue, we're going to have to, and the tens, if not hundreds of thousands of students and faculty and staff that we've got, this is exactly where we have to get to in terms of economics. Both the time frame and the economics. We're looking at buckish a test because we can't afford it otherwise.
Yeah, no, thank you. Yeah, we're really excited. I mean, we started dabbling in this just recently, but the data look really promising, and it would be nice to get something out there to help people.
And this is exactly why our office helped to organize a webinar, so that you get more exposure to the corporate and investor community as you're looking for capital strategic partnerships to help commercialize all that great technologies. Great, thank you. Thank you, Dan. Now, I'm going to turn over to our next presenter, Dr. James Friend from Respite, UC San Diego Mad Lab.
Thank you. Let me get everything going here. All right. Hello, everyone. Thank you for your time. I'm going to be talking to you about Respite, a 1-minute testing idea for COVID-19. And let me begin with kind of a brief note about our group. We have a PhD student by the name of Shuai Zhang, who's a acoustofluidics person and experimentalist. The acoustofluidics aspect will become clearer as we get into this. Philip Kyriakis, who's a diagnostic expert and experimentalist as well. Cecile Fleurer, who's a postdoc of mine who works on acoustic devices. Todd Coleman, who's from bioengineering, and quite a few people probably know him. He's from medical technology and testing. Myself, where I've worked on medical devices and acoustofluidics, I guess, longer than I'd like to admit, over 20 years, I guess. And I've co-founded a number of startups and had a few startups over the years by my students. Professor Rob Knight, who's a star in biology and who has students right now crawling through the sewers of various cities looking for COVID-19 as a diagnostics person. And our particular work is associated with Matter Labs and Brian Wendt. This particular company from Ventura, California, is a small company, but it specializes in high technology translation from the lab to commercial benefit. So in any case, the problem, as you no doubt know, is asymptomatic COVID-19 carriers produce the unchecked spread of infection. And so really for modern society to work, we've got to get our hands on who these people are and for them to be quarantined. And really for sports events and everything, all these things we took for granted from before are impossible today, as you know. Unfortunately, the fastest test today is 30 minutes. I'm a gene expert by CFID. And really, you know, how many Americans can wait 30 minutes? And when you stack these tests together, then inevitably we get behind. And so you have to wait until later to find out your data. The truth is that the typical speed of testing nowadays is on the order of 24 hours. And so what ends up happening is you get the test and then you find out by phone later, sometimes up to 14 days later, which is just a waste because, you know, clinically irrelevant. And if you consider, for example, you know, earlier was brought up, what about the university environment? But you say, for example, a typical venue needs 1,000 tests a day. If you have 30 minutes per test, that's 500 hours. And so then if you take, well, there's 24 hours in a day and take that 500 and divide by 24, then you get about 30 parallel diagnostic lines are needed just to get those tests done in a single day. And this really is the problem with all this testing at hand. And then, you know, I'd like for you to imagine then, what if you could accomplish the test in a minute? So then that 1,000 tests becomes 16 hours with 1 line and 1.6 hours with 10 lines. And say, for example, you know, for a flight or for a sports event, something like having a number of parallel lines is not out of the ordinary. And while you wait, venue testing then becomes possible. Furthermore, there's recent published work that suggests that contact tracing could work to essentially end the pandemic by reducing R0 to below 1. Most importantly, you know, it saves about 1 million person hours per day across the US to accomplish this. And so the way that we're proposing to achieve this is by using acoustofluidics, where we use a very high frequency 100 megahertz acoustic driven pumping and mixing phenomena with lateral flow immunoassays, just as like my colleague Dan Kamei from UCLA was talking about previously. If you take, for example, the wicking material that is present in one of these LFI assays and then just let it sit, then, and when we put in a fluorescent dye, for example, here is shown in the standard row, then what you'll see is that, you know, the fluorescent dye is there and maybe we have a wicking pad on the outlet. Then we introduce a second fluid. Here it appears a bit purple, not fluorescent. And after, you know, 40 seconds or more, the flow actually completes through, and essentially you have that fluorescent fluid is actually completely gone. That's the time that it takes for this fluid to flow through is a little while, you know, a few minutes. With acoustofluidics, we're actually pumping the fluid through and we see essentially the same thing happening in less than 20 seconds. And so, you know, I mean, how is this any different than a lot of the other tests being promised out there? What we're going for here is the recognition that diffusion must occur for the target in a sample to be accomplished for the to have binding for the antibodies for detection. And this is where the bottleneck is actually occurring. And so really there's no effective means to reduce the diffusion time that has been found so far. And what we've done is using these high frequency acoustofluidics, we're seeing that we're able to reduce the diffusion time by on the order of 3 orders of magnitude. In other words, if you say something's going to take 30 minutes, that becomes 18 seconds to achieve the same outcome. And furthermore, this also is promising to improve the sensitivity of the LFIA by at least 1 order of magnitude, probably 2. We come from using this technology in lithium metal batteries where they're not rechargeable. But we've shown that you safely can recharge lithium metal batteries at very high rates as a consequence of using this technology by overcoming a diffusion problem in that technology. And of course, as Dan, you know, mentioned previously, you know, LFIA tests are old. They're dated from the 1960s. They're inexpensive, just made out of paper or polymer material. They're trusted as point of care tests of pregnancy to heavy metal detection. And they work with blood, saliva, and other things. And the LFI market is very large, 8 billion, and pre-COVID CAGR of 7.6, and God only knows what it is now with COVID. So our embodiment, what we're talking about doing here is this. As it turns out, we use the LFI tests as our blades, and then our we call it a speed reader as the razor in this arrangement, where, if you will, in this box, this black box here, we have the acoustics technology that enables the LFI test to actually proceed much more quickly. Now, the LFI material, the wicking material, needs to have it has some special properties to it to make this actually function properly. And so it combines the proprietary LFI wicking media with this technology as sort of this razor and blades idea. And of course, the razor and blades model, the whole concept behind it is this idea of having, you get the speed reader for free, and the blades are just a little bit more expensive so that you can cover the cost. It's worth noting the LFI is already out there to test for COVID-19, mainly from blood. There are 4 LFI tests that are listed here, and our aim here is to actually, to make this technology quicker rather than compete directly with what they're doing. So our development plan, I said it's simple, but looks like arrows have been spilled onto the page. I'm sorry about that. We've got the initial concept. We've had some gratefully received some funding to try to de-risk the acoustic wave device, combining there with the LFIA tests. And we filed the intellectual property today, in fact. And next step is to test with mock COVID and bench prototype. We can get our hands on mock COVID from Rob Knight. And then as well, we can get human samples from that lab. And an important part of working with human samples is that we need to do the IRB and rely on what's called the PrEP Act to let us pursue that. That's actually not going to be really a problem. We're going to need some additional funding then to make this work. Where everybody, else comes into play here really is a manufacturing partner, and to get us set up for the emergency use authorization filing and the final prototype development. At that point, we pursue either a spin out or licensure, depending. Frankly, I don't care. I mean, the whole point of this is that we're trying to save people's lives. And whatever works to make that happen is good by me. But the point of this is we want to get to limited mass production and at least testing at UC medical centers, relying on the PrEP Act to get us to that point. If we get sufficient data, then we're able to potentially file the 510K via the Q submission system at the FDA. And the purpose is to recognize that LFI is used for a lot of other technology, a lot of other conditions as well, cancer, metabolic diseases, forensics, and we'd be able to speed those things up. And so then, it has a much broader market after presumably and hopefully COVID-19 is done. But likewise, we need to do large scale mass manufacturing to get us the mass testing for COVID-19 that everybody wants to have, and for the rapid testing. Our intellectual property position is, we have a novel acoustic device that provides the design features actually to make this work. And then we have the novel using LFI. And then there is particular characteristics of the LFIA test media that we need to make the acoustofluidics work. And combine them together, and we've got this package to make this happen. So from here, initially, the funding is sufficient thanks to the UCOP. More funding, of course, would facilitate more rapid progress, especially as we go from the mock COVID to human samples. And our milestones, in the near term, are to de-risk to test with Cuban COVID-19 samples, and then to get sufficient data for the FDA emergency use authorization filing. That is, if the emergency use authorization is still in force. I have filed for ventilator under the EUA, and I would say that experience is not necessarily all entirely positive. But that being said, it should probably be done by a medical device manufacturing partner and a licensure model or some sort of spin-out function. We do need some strategic partners at development to production, because that's not what we really do. We need to see about translating it to clinical use beyond UC-affiliated healthcare facilities. There's just that market penetration into those facilities elsewhere across the U.S. and indeed internationally. It's not me and it's not my team that we have together now. The FDA emergency use authorization, 510K filings, and all of that's coming needs to be done by a medical device manufacturer, hopefully that's partnering with us or licensed the technology out. And they need to be registered with the FDA, and then of course in compliance with all the ISO requirements and so forth. we need their help with that. And with that, I'll finish, and I welcome your questions.
I was on mute. Okay, great. Thanks, James. Doogie and Christian?
Yeah, James, first of all, congratulations on the team and the progress so far. Very, very impressive. Just a couple of questions. One of them is there any mechanism for detecting viral load in this case? And then second, what are you hearing from the manufacturers or any downstream partners in terms of timeline and costs involved in prototyping and so forth?
Okay, well, I'll answer your second question first. Downstream partners, we do have, a number of companies that are interested in this that make LFI tests, but they want to see the risk lowered. And obviously, because LFIA is not particularly necessarily a risk, but what's this acoustic stuff? And so that part, I think they need some convincing. But I think in a month or so we'll get there. This is kind of a mad rush project, really, due to the circumstances. And then I think they might very well pick this up again. My hope is, we are able to span that valley of death. As far as the viral load is concerned, I think, with these tests, the whole aim is either there is a viral load that is detected by the sensor or not. And we don't have the data like, Professor Kamei has at the moment to say, whether the LFA test is going to, detect every potential case of infection or not. I wish we did. Our main focus has been, rather than though we are making the LFA tests here, is to make them faster rather than, addressing that specific instance at the moment. Because the thought is that at some point, we'll fix that problem, but we just need to, 30 minutes is just not going to be good enough.
And I think just following on that, James, I think you're absolutely right. The diffusion, the latency and the time has just got to be much faster. As you say, people just cannot wait. We can't be sending people away and just not knowing. And so you're going in exactly the direction that we know we need to go in terms of the turnaround time and that latency. And the more you can bring it down to something approximating near real time, whatever that might become as an industry standard, we'll have very strong support. But this looks great. Thank you for this.
Thank you.
Great. Thank you, James.
Thank you all.
Great. Now I'm going to turn it over to our final presenters, Dr. Wisher Wirtz and Dr. Lanham Merman from uPlasma, UCLA.
Okay. I'm going to share my screen here. All right. And you can all see that. Let me put this into presentation mode so it looks a little better. All right, are you seeing my correct screen, or do I need this?
Yes, we're seeing your screen.
Okay, so you can see the presentation. All right, great. So what you see here is a beautiful picture of a plasma, all right, the fourth state of matter. And it's the source is on the left and the plasma is shooting to the right. And in this case, we're looking at a plasma that is disinfecting a surface on the right. So I'm going to talk about this. This is where uPlasma was formed, and Layla here on the call is the CEO, and I'm the original founder of the concept. And what we're going to talk about is the merits of plasmas. And just like the other presenters, uPlasma is this idea we want to get plasmas out to everyone, and so that everyone can use the benefits of plasmas for disinfection. So the idea behind uPlasma is that we want to provide an air plasma solution for everyone, so that we can avoid the burdens of hospital acquired infections and even worse now with the COVID-19 pandemic. And the key to this is just a summary slide, but the key is that we have a patented technology that, again, is scalable, and the key here is affordable. Okay, there definitely are plasma devices out there, but what we're trying to do is to make a plasma device that is affordable. and still has the microbial inactivating power of a plasma. So, and an important thing here is that it's higher efficacy than standard sanitation techniques, no toxicity, and no side effects. So current disinfection methods are primarily chemical. And of course, we're all familiar with alcohol-based and chemical disinfectants. And you've heard some about UV is an example. Some people often compare plasma sanitation to UV. The problem is UV, you really can't expose yourself to UV, right? It's inherently dangerous, indiscriminately kills everything. Plasma actually will only kill viruses and leave healthy cells alone. And we'll talk about why that is. So looking at the list of benefits of each of these methods, we definitely have chemical is very scalable, and that's the reason it's used. You can put it in everyone's hands. But the problem is that the efficacy and the cost is all show. Inactivating agents also have their challenges. And then cold atmospheric plasma, we're working off the premise that cold atmospheric plasmas are beneficial, but we have come up with some solutions to the cost, the scalability, and the operating conditions, as I'll show. So first of all, why does it work? So cold atmospheric plasma could be used to sanitize your hands, any surfaces, or even to sanitize the air. And there is much information, many results from many researchers about plasma effectively inactivating bacteria and viruses, and even killing cancer. And that's a lot of what we do in our lab. So how does it do this? Well, what's interesting about plasma is it takes the air, think of air as oxygen and nitrogen, and it turns it into reactive oxygen and nitrogen species. These reactive species can then attack a virus, and they can break the cell wall, they can attack the DNA and RNA, and they will preferentially, again, attack the DNA and RNA of faster replicating things like viruses and cancer cells. And also can, one of the key mechanisms is the amino acid oxidation. So you can tune your plasma to kill in whatever way you want, to kill what you want and to leave alone what you don't. So what's our key at uPlasma? Again, we are leveraging, we're actually going on our second decade of people realizing that plasma has these benefits. The problem is that people use these custom-made electronics and they use noble gases. And that alone is logistically a challenge and also drives the cost into the thousands of dollars per devices to tens of thousands of dollars. And that's because of highly customizable electronics. But for those of you who do research, you know that you can just simply go on Mouser or DigiKey and get really low-cost electronics to do the same thing. Plasmas are pretty amazing state of matter, but it's actually quite easy to create. So we use low-cost electronics to generate these plasmas. And so what does that lead to? Well, it deactivates the virus. And we've been showing this. We did a test on Friday of deactivating the virus, and it just completely obliterates the virus. And that's because these reactive oxygen and nitrogen species will go in there and attack all of the viruses, and we get increasing efficacy as we go. Now these are shown pictures where we're deactivating the virus in a solution, and that's actually going to take a little more time. But we've been able to deactivate a dry virus in under 15 seconds. And our particular plasma is actually faster because it has higher reactive oxygen and nitrogen species content because we don't use the noble gas to seed the plasma. So what does that lead to? That leads to basically a low-cost device that only needs to operate on ambient air. And it's something that you could have wall mountable and you could sanitize your hands, for example, or any surface or any surgical instrument, that sort of thing. And you don't have to worry about, as you all know, we walk around and we go to a pump station and it's out of the sanitizing gel, right? But we don't run out of electrical power or ambient air. And so what that leads to in our business model is our first business model is for hand sanitizing and surface disinfection. And it's a business-to-business relationship where we want to go to hospitals, primarily in healthcare facilities, right, to prove this efficacy and to get, just like all the other presenters, we want to attack the COVID-19 pandemic, and we think that this is the most urgent matter, is to get this into healthcare facilities, prove out there, and then move on to our other use cases. So to do that, we have had many accomplishments from submitting the provisional patents, forming this company, and developing a UCLA healthcare partnership, and Bell Labs is also our partner for manufacturing and sales. But right now we're at this valley of death, right? We're looking at it, and we're looking at around 300K to move us over the next about half a year into getting to doing critical tests and developing prototypes that we can hand to customers and even have them use. For example, on this slide, I have the BSL-3 lab, which is the biosafety level 3 lab here at UCLA, where we've been actually able to kill the SARS-CoV-2 virus. And they actually want our device because we've shown that, unlike other devices, we can kill on cloth surfaces, cardboard surfaces, these types of surface organic materials that other people have not been able to kill the virus on effectively. So looking at that, we want to move forward as quickly as possible, get our devices out there to healthcare facilities, and then move forward. And this 300K that we're talking about is really just to get our organization moving in the right direction with a commercial prototype that is actually something that people are using every day. And looking at the cost, if we want to kill the virus, but why use this instead of something else? Well, if you look at sanitizing gel, you have to constantly fill it. And looking at the average cost for a station, you're looking at about $300 dispenser, and then you have over $600 in average cost for the gel itself. So now you're looking at $700 per year plus labor, whereas this is a device that you can just mount on the wall and it's less than $100 per year. So it is cost effective. It sounds like science fiction early on, but the idea is actually we're leveraging that science fiction to bring lower cost and higher efficacy. So here's our team. As I mentioned, myself and Layla and Jitong Chen is our brilliant plasma medicine postdoc that has been working on this for almost a decade now. And we have been developing devices, like I said. We were killing cancer, using it for dental applications, and when COVID-19 came out, we said, okay, we have to address this. So that's why uPlasma exists.
Great, thank you, Richard. Doogie and Christian?
Yeah, hi, Richard. Great job on the presentation. I thought you really spelled out well, which was going to be my first question, is why you're starting with the hand sanitizer market. I think you did a great job of kind of analyzing the cost variation on what's already in the market. You mentioned, so two things I'd like to know a little bit more about, some of the other areas that you see this application working for in terms of materials and services, and then also related to what you need outside of capital to help you move this along.
Right. Yeah. So if outside of capital, I think what we do need is those partnerships, right? And we need to get with our healthcare and hospital partners, which we've already done on small scale. But if we're not doing this for many partners, and there's a lot of use cases and a lot of data that show, and I should just say results from customers showing that they're using this and that they're putting their gel hand sanitizers aside and using this plasma. That's what we want to do. So that's why we think this forum is fantastic, because we can reach out to people that would say, yeah, I would want one, right? And I think that that's I think the initial capital is for us to develop that initial prototype, but I would say on top of the 300K would be a partner that would be willing to make prototypes that we can send out essentially to free for our initial partners. Does that answer your question, or did I miss any aspect of that?
It does. The only other part was other other use cases.
Oh yeah, definitely. With other use cases, actually, that's a great point. One of the things is we found that surface contact is definitely an issue. However, the airborne is another issue, and you can actually use this same plasma device to clean the air, right? If you're in a recirculating environment, you can actually purify the air with these plasma devices. And it's more effective than other devices because we can actually tune the parameters so that we're just killing the virus and we're not introducing say ozone to the environment.
Thank you.
So Richard, if I may, and firstly, that was great. That was really helpful. Thank you. I think about one of your penultimate slides where if you think about the efficacy and you think about moving from, greater than, the uPlasma being greater than 95%, which is far more effective than the sanitizing gel. There's a little bit of a chicken and egg here, I guess, of how do you get enough data validation for you to make the shift? Because as I think across our system and our many hospitals, we're spending many millions every year now, and it's just gone up a lot on sanitizing gel and hand sanitizers. And so we would probably want to make the shift because we could see the absolute advantage both clinically and economically, but there's the how do you give the data evidence before you actually do it? What's your plan? Because we certainly fit within, I think, one of your core customers, but there will be a degree of not resistance, but just reticence until they can see more of that data evidence. Do you have a way of kind of breaking through that tension?
Yeah. So thank you for that question. Yeah, we have actually been in the BSL-3 lab killing the SARS-CoV-2 virus with a virologist on many different surfaces. I actually used the New England Journal of Medicine paper that talked about plastics, stainless steel, copper, and cardboard. And I actually used Amazon cardboard boxes and put it in a 6-well plate. And we effectively killed the virus on all of those surfaces. And more recently, my virologist is a sports fan, and he wanted to see it killed on sports equipment. And so we did footballs, baseball, and basketballs that are used by the NCAA and the NFL. And so we looked at that as before we actually do in vivo or actually clinical trials on people's hands. We saw leather as an organic surface, and we just got the results back last night that showed that it did effectively kill the virus on those leather organic surfaces. And so you're exactly right. The next step is for us to hopefully kill it on an actual living skin. Does that answer your question?
No, it does. Thank you. That's definitely I think the stages we need to get people comfortable with a shift from something they can see, and there's always that confidence that comes from what people can see and touch, even though they might be misplaced.
Yes, that's an excellent point. And we do have methodologies that we've just patented that would allow there to be a little bit of a feedback loop as well with it. So we're very cognizant of that we are dealing with humans ultimately, and not only do they want to be clean, but they want to feel clean. Right, and I think you're right that's very important and something that we are going to work with people to see what gives them that, not only the numbers that show that they're clean, but also the feeling that they actually did get the right treatment.
Great, thank you, Doogie. I see that we have a question in chat, but we're running out of time, so I'm going to connect you offline to answer that question. So and that concludes our showcase today. I want to thank our panelists as well as our presenters for sharing your technologies and teasing out further details. So the recording is going to be shared in the post-event emails. Please reach out to the presenters if you'd like to learn more. Thanks again for joining, and I hope to see you at session 2 on Wednesday. Thanks, everyone.
Thank you.
Thank you.
Thank you.
All right.